All right, this is next segment, part 2, on the citric acid cycle, among other things. Citric acid cycle, or Krebs cycle, happens in the mitochondrion, and you'll see why in a bit, but oxygen is required for this process to begin. It happens in the matrix of the mitochondria, which basically means the middle, the inside. Mitochondria has a double membrane again. So there's the matrix, and then a membrane, and then the inner membrane space, and then the outer membrane, if you like. Before it can begin, we've got to take the pyruvate from glycolysis and bring it into the organelle, into the mitochondrion. And that's done using a transport protein, name not important. You'll notice here pyruvate again is 3 carbons, 1, 2, 3. And as it enters, we have a reaction where we lose a CO2. Here's our first CO2 that's given off, which is why we breathe CO2 out. And then it also the pyruvate gets oxidized as we reduce NAD plus to NADH. And again, this NADH becomes important in the electron transport chain later. And we combine the remaining 2 carbons with coenzyme A to form the important point here, acetyl-CoA, or acetyl-CoA, CoA standing for coenzyme A. And here we have a 2-carbon molecule. Here's the overall process, and you'll notice that the individual names and enzymes and so on are not the important factor here. The general way we like students to understand this is based on carbon number. Citric acid cycle is basically rearranging carbons cyclically, constantly adding in a new 2-carbon molecule to generate electron carriers and ATP. Here we have our pyruvate. We already showed you we made NADH and CO2. Here's our acetyl-CoA. We add that into the citric acid cycle. During the process of all this rearrangement and energy production, we lose more carbon dioxides. We make additional NADH and FADH2, and we also make some ATP. Remember that we had produced 2 molecules of pyruvate from glycolysis. So 1 molecule creates 1 turn of the cycle. So from 1 glucose molecule, which made 2 pyruvates, we'll actually get 6 NADHs, 2 FADH2s, and 2 ATPs. And as a little side note, the initial reaction here doesn't actually produce an ATP. It actually produces a GTP, G for guanine, which then gets converted into ATP. Here's the specifics of the reaction. Again, not important. Acetyl-CoA added to oxaloacetate. It's a 4-carbon molecule to make a 6. We rearrange it, blah, 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 and we get back to our 4-carbon molecule. Then we enter oxidative phosphorylation and chemiosmosis, which is basically using an electron transport chain to take energy from electrons, from charged electrons, and slowly release it to power a pump, and that pump produces ATP. So those electrons came from glycolysis and the Krebs cycle, and they're being carried as NADH and FADH2. These 2 electron carriers will donate the electrons to the ETC, and they power the oxidative phosphorylation. So here we see this stepwise process. You'll notice NADH adds its electrons here at the beginning of the chain, and FADH2 adds its electrons here. We don't need to know all these individual stepwise things. That's not the important component. One thing to notice here, however, is that NADH, because it adds in its electrons earlier, NADH allows the cell to produce more ATP per NADH than does FADH2. So at one point you're going to see me say the electron transport chain, or oxidative phosphorylation, produces between 32 and 34 ATP. Well, what's the cause of the difference? It's whether NADH gets produced or FADH2 gets produced. And we'll show you that in a bit as well. The second thing to notice here is that the electrons, once they've gone through this stepwise process, this ETC, the electrons are still there. They have to be absorbed by something, and they get absorbed by oxygen. Okay, oxygen gets reduced. We then add the protons, and we make H2O. And this is how respiration makes H2O. It's because of the electrons that were created during the catabolic breakdown of glucose. Here is this ATP synthase that I mentioned previously. This is chemiosmosis. And I like to think of this as one of those baseball hats with the spinning fan on top, but it's upside down. Here's the spinny fan down here, and here's the part that's on the head. And what we will do is create a gradient using protons. Here you see the intermembrane space, and here's the mitochondrial matrix. This is where the Krebs cycle occurred, and in this membrane here is where we had the electron transport chain, passing from protein to protein to protein, these electrons. And what that energy is used for from those electrons is to pump protons against their concentration gradient to the intermembrane space to create a very giant concentration gradient that then will allow these protons to flow through the ATP synthase and cause it to spin. And as it spins, it phosphorylates ADP into ATP. It provides the energy. So as I just mentioned, the electron transfer through those proteins helps to pump protons from the matrix to the intermembrane space. That's the space between the inner and outer membranes of the mitochondrion. And this creates a proton gradient, as we said, which drives the chemiosmosis of the ATP synthase. And some people call it a proton motive force. Here's the overall process, as I mentioned, and this slide is a super nice representation of what we're looking at. This is the intermembrane space. So out here, what we can't see is another membrane on the outside of the mitochondrion. And here's the mitochondrial matrix, the internal component. So in here, we have the Krebs cycle occurring and producing NADH and FADH2. Those two electron carriers add their electrons to these proteins, here 1 and Q, and cause a conformational change in these proteins that moves a proton against its concentration gradient into the intermembrane space. As this builds up, this continues to occur. The electrons, as they remain at the end, get added to oxygen to make water, and then this proton motive force, if you like, chemiosmosis, passes through the ATP synthase to phosphorylate ADP into ATP. And we call this chemiosmosis, plus the electron transport chain you saw here, both together is oxidative phosphorylation. We already talked about previously that glucose, as it gets broken down, makes NADH, which transfers its electrons to the ETC to make ATP. And here, one thing to notice, where we talked about, if you look over here where it says oxidative phosphorylation includes electron transport and chemiosmosis, these processes will make 32 or 34. And the or is from over here. When glucose gets converted into 2-pyruvates, we make 2-NADH. Well, as this NADH goes into the mitochondrion, it either will stay as NADH or it will get converted into FADH2 by these electron shuttles. And if it's FADH2, we'll only get 32 ATPs. If it's NADH, each NADH will give us an additional ATP because it adds its electrons earlier in the chain. And as a result, we would get 34. So again, review here. Glycolysis makes 2 ATPs through substrate-level phosphorylation. It occurs in the cytoplasm. Krebs cycle occurs in the mitochondrial matrix. Begins with 2 acetyl-CoAs, which came from the 2-pyruvates, a 3-carbon molecule converted into a 2-carbon molecule. 2 ATPs are produced, 1 per turn, through substrate-level phosphorylation. We also make additional NADH and FADH2 to bring electrons to the electron transport chain, which will create a proton gradient inside the intermembrane space, which will then power the ATP synthase, and through chemiosmosis produce between 32 and 34 ATP. So about 40% of the energy from glucose gets converted into ATP, and in the end, in total, adding glycolysis, and it's net 2, and adding Krebs cycle, and it's net 2, and adding oxidative phosphorylation, and it's net 32 or 34, we get a total of 36 to 38 ATPs from 1 molecule of glucose. And on that note, we will take a pause.